Okay, hello everyone. Today I'm going to discuss about ABO blood group system. Today topic is ABO blood group system, and previously I have discussed about the introduction part of our blood bank and the transfusion medicine. I mean the transfusion started. That is we have already discussed. So today the ABO blood group system and the antigen and antibody, those things and all I'm going to discuss. So the class code is, course code BMLS3005 and the course name is blood bank. Okay. So on the screen you can observe this RBC cells, and this side, another side is the antigens, right? So on the RBC cells, on the RBC cell surface, this antigens it will be bind. So before this antigen, even this precursor substance, this H antigen, it will be bind on the RBC cell. Okay. So you can see on the RBC surface, first of all, this RBC surface, this H antigen it will be bind. So that is a precursor substance. On the precursor substance, it will be bind either A antigen or B antigen. So if it is a both, it will be add on the RBC surface like a one side here and second side here. It means that AB blood group. For example, on the precursor substance, if it is added A antigen means A blood group. If the B it is added, so B blood group you can call it as. So like that. So it is a this type of this antigens. It is a nothing but a sugar zone. So what are the precursor substance sugars? You can see there is a galactose, galactosamine and N-acetyl glucosamine, right? These are the and with the complete form of your precursor substance should be added glucose sugar. So this is the main thing, that is the antigens it will be present on your RB cells. Okay. So now come to the point, the introduction part of your blood group. So blood grouping is based on type of antigen present on the RBC cells. So that is already we have discussed in the previous class. That is on the RBC cell it should be present. So there are more than 300 blood groups systems, ABO and RHA, are of importance from the clinical point of view. So clinical point of view, we can discuss about the ABO and RHA system and all. So other blood group systems are MNS, Lutheran, Lewis, and the Fekin. Those are the, even though 20 blood group systems and all, it is in the universally in the there. But the thing is, the ABO and RHA system, clinical point of view, we are using. So discovered by the Carl Landsteiner in the 19th century, and red blood cells that contains different types of antigen, that is the agglutinogen, and while the plasma contains the antibody, that is the antibody agglutinins, we can call it as genes that controls the system are present. So Carl Landsteiner, what he states that if an antigen is present on the patient's RBC, the corresponding antibody should not be present on patient's plasma under the normal condition. So normal condition, if any antigen, for example, you can take it like a antigen on the presence on the patient's RBC surface. So the corresponding antibody means it is the same antibody. I mean that A antibody should not be present in the patient's plasma, because of if it is the same antibody it is present in the plasma, what will happen is going to be clot, and is the patient is going to be death and all, it will be occurs, right? So next slide you can see this one, it is a major ABO blood group system. So the patient is having the A blood group means the antigen is presence A. So antigen, what is the antigen is missing here? B antigen is missing. And antibody, what type of antibody is present here? Antibody B. So at the same time come to the B blood group. So for the B blood group, antigen B present and antigen missing one is A. So antibody A, it is a present in the B blood group. So it is a reversely you have to think about it. Okay. So when you are coming for the O blood group, no antigen is present here. And what type of antigens are missing here? A and B antigens are missing. And what type of antibodies is present here? Because of here there is no antigens here. So it means that the antibodies it will be present in the patient's plasma. That is the A and B. So come to the AB blood group. So the antigens A and B, it is a presence, and the missing antigens are none, because of though both are, it is a presence in the RBC surface, because of no missing at all here. So antibody, so no antibodies because of already the antigens are present in the patient's blood. So now come to the point, methods of blood grouping and all. So methods of blood grouping, slide method and tube method. Slide method and tube method, right? So tube method is a better method and but takes a longer time. So takes a longer because of that that incubation period and all, it will be differ from the slide method. So okay, simple in a tube with the antiserum, incubate it, the centrifuge it and examine it microscopically for the agglutination. So what are the requirement for the slide method? What you have to take a 3 slides, antisera A, B and the blood samples you have to take it. And procedure, take a 2 clean slides and mark it 1 and 2 and put 1 drop of antisera and A and slide 1. That is you can label as a 1. And 1 drop of antisera B on 1 slide 2. That is you can label as a 2. So add 1 drop of blood to each and mix it well with the stick. So wait for the 5 minutes and observe the result. So how you can going to observe here? If an agglutination occurs, it is visible with the naked eyes as a dark reddish clumps of the different sizes. So if agglutination is a minimal, it can be confirmed by the examining it under microscopic and all. So like that. So you can see on the slide there is a observation should be like this. The agglutination it is happen in this slide and there is no agglutination here like this. Okay, the observation should be like this. So interpretation you can give like this: agglutination with the antisera A not with the antisera B. That is a group A. Agglutination with the antisera B not with the antisera. It means that is a group is a B blood group. Agglutination with the both antisera A and B, that blood group is a AB. No agglutination in any slide, that is we can call it as O blood group. So it is the agglutination one, the reacted and non-agglutination. Universal blood donor is a O blood group because of no antigens presence here. And universal receptor blood group is a AB, as both A and B antigens are present. So agglutination occurs in both, has no antibodies present in the serum. So now it is a Bombay phenotype. In this, it is a here I am going to give the brief introduction about the Bombay phenotype, and in the further classes I will discuss more about it. Okay, very rare, and H is a precursor substance of antigen A and B. So this people lack of H and along with the A, B antigens, but the plasma contains A and B and H antibodies. And Bombay phenotype are individuals who lack the H gene phenotype, that is a Rh positive, that is the antigen you are finding, and these individuals constitute that is the 80% of the population. So when you are going for the Rh negative, that is there is a options of D antigen. These individuals constitute of them, that is 17% of population. These are the references you can go for this and you can study about it. Okay, thank you so much.